Thank you. Oh, yes. Thank you for the introduction. And I'm very glad to be here. And also thanks to Sarahi and Rachel for inviting me to give this talk. I'm going to say I'm delighted because so earlier this year we were on quarantining before an oceanographic cruise, and I spent several afternoons watching these videos that are very kindly posted online, and I saw like the very high level of the talks and the nice, very nice lectures that you have had, guys. The opportunity to see and learn from. So it's a real pleasure to be part of this. So right, let's go to business. I'm gonna talk about nitrogen fixation, which is, as you said, is my main topic of research. And what I would like today to present is that how can we go a little bit beyond our current knowledge in nitrogen fixation. You may be familiar with this process. This is a microbial process that takes place in the ocean, but not only, also in soils and in other environments. And in the ocean, it has been much of our knowledge stems from subtropical and tropical regions, and also from only a type of organisms, mainly the cyanobacteria. So what I will try to show you today is that this is also happening beyond the tropics, and is also being done by other types of organisms, not only cyanobacteria. So, oops, there we go. So just to start with the basics, nitrogen fixation is a biogeochemically important process. Just looking at how the process works, nitrogen, it's part of our atmosphere, and it's dissolved into seawater, and the isotopes can take it up and reduce it into ammonium. So ammonium is then more labile and easily accessible source of nitrogen for the phytoplankton community. So this is basically a source of nitrogen for phytoplankton communities, which eventually can transport that organic matter with them into the deep ocean, which is then going to be remineralized into nitrate. So overall, this cycle that is being started by diisotopes is actually a main source of bioavailable nitrogen for marine systems. But where is nitrogen fixed? Well, if you look at the stoichiometric reaction here, you can see that this is a very expensive process for the cell in terms of energy. So for every molecule of nitrogen that you fix, you're going to need at least 16 ATP, and that varies as well with the type of enzyme you're using to do this. This means that in general, our classic belief has been that if there is nitrogen available around you, such as nitrate or ammonium or amino acids or other typical bioavailable nitrogen forms, why would the cell dare to fix nitrogen that is so expensive? So in general, if we look at the distribution of nitrate in the oceans, you see that much of the open ocean has mostly undetectable nitrogen concentrations. So these are the places where we have always believed that nitrogen fixation was going to be more important, which coincides with the places that are also poorer in biomass. So this is the distribution of chlorophyll. So in all, it is estimated that nitrogen fixation provides up to 50% of new production in the subtropical gyres in these great oceanic deserts. If we put it in a budget as well, nitrogen fixation globally provides the largest source of reactive nitrogen globally, above atmospheric deposition and also riverine input. But all these inputs are actually counteracted by other microbial processes and physical processes that can remove reactive nitrogen from the ecosystem, such as the nitrification, anemox, and other nitrogen loss pathways, and also sedimentary organic burial. So there are different estimations of both these sources and the sinks, but no matter which estimation you use, you always come out with a negative balance. So this can mean that either we are not measuring nitrogen fixation properly or any of the other processes, or also maybe because nitrogen fixation is being done in other places that we are actually not taking into account. And this is the way I would like to, the message I would like to convey today. So this is the global distribution of nitrogen fixation rates in the ocean, updated in 2019. and there's going to be an update. It was meant to be this year, but it will probably be early next year. And just at a first glance, the thing that you see is that most of the measurements have been done in the North Atlantic, that there are a few in the South Atlantic, and the rest is pretty much very sparse. With some time series stations that have a lot of measurements, and practically nothing in the Indian Ocean and nothing in polar regions. So this is already a very biased geographical view of the process when we try to understand its global impact. However, some adventurous people have started to measure nitrogen fixation in unexpected places. So for instance, this is the eastern coast of the USA, where they found very high rates in the shelf area. Also in the Arctic Ocean, and this is a very active topic of research nowadays. And also in the Antarctic Ocean, there have been found some quite high rates as compared to what we typically find in the open ocean. So if you put together all these different unexpected places and you put it in a global scale, you come up with some about 20% more, which will still not solve this imbalance, but would at least make it a little bit smaller. However, these measurements in unexpected places are still quite sparse and not done like by default in time series, in transect monitoring programs, and so forth. So it's hard to assess whether this 20% is real, if it's actually more, or if it's actually less. So, but it gives us a hint that something is happening beyond our typical area of research. Also, when we look at models, so this is, for instance, the outcome of a species distribution model towards the end of the century. Models like this predict that the isotopes are going to extend towards polar regions. For instance, here you can see that we expect an increase in appearance of new diisotope groups in higher latitudes. So this is something to worry about how are diisotopes going to impact higher latitude ecosystems in the future ocean. But who are nitrogen fixers? So nitrogen fixers are called diisotropes, and this comes from the Greeks, where D is 2, azo is nitrogen, and trophos means nutrition. So these are literally the bugs that feed on nitrogen. And as I said in my very first slide, typically we have focused on cyanobacteria. And this is because before the development of molecular techniques, when we could only count on microscopy, we will only see things that are big enough to be seen on a microscope and identifiable on a microscope, such as filamentous cyanobacteria, such as Trichodesmium or Nostoc, or some unicellular ones, such as Crocosphera. But when you take into account all the published sequences, and this is looking particularly at the NIFH gene, which encodes for a subunit of the nitrogenase enzyme, we actually see the cyanobacteria just are a really tiny part of the story. So there is all these other groups, all these other clusters that are other non-pigmented bacteria and also archaea that are actually more prevalent or majoritary in the overall picture. And because we don't have a better name for them right now, we just call them non-cyanobacterial diisotropes, or NCDs, because they are just everybody else besides the cyanobacteria. So I'm going to go a little bit deeper into our current understanding of these NCDs. So the past picture was done with NIFH amplicon sequencing, but just quickly to say that if you take metagenomes, and these are the metagenomes from the Tara Oceans expedition, you pretty much find the same picture. So you have that NCDs are actually majoritary as compared to the cyanobacterial diisotopes that are less prevalent. However, one of our main challenges nowadays is that these organisms are not available in culture. There are very few available in some collections. So for instance, this alpha protobacterium has been isolated by a group from MPI in Bremen. And this is a pseudomonas that has been isolated from the Baltic Sea, and it's available in the University of Copenhagen. But this is certainly not representative of this huge diversity that we have seen in these phylogenetic trees. and that refrains us to go deeper into a more deeper understanding of their metabolic functioning, the physiology, and so forth. But so another current effort that has been done by some collaborators of ours from the University of California, Santa Cruz, and they put together like all the NIFH amplicon sequences published thus far, and also the metagenomes. And basically there are two main clusters that stem out from this. And one is the gamma protobacteria and the other one is the delta protobacteria. And something that we are starting to see is that actually gamma or delta NCDs, they have different distributions in the ocean. So for instance, this is an expedition in the southern Indian Ocean, the Sphinx cruise, which is a geotraces program. So this is the tip of South Africa, and this is Madagascar, and this is the transect of the cruise. And stations are pulled by temperatures. So in this case, we found that, for instance, cyanobacteria such as Trichodesmium, they were dominant above 20 degrees. But if you go to lower temperatures, so towards the southern part of the transect, you find mostly gamma protobacteria, which seem to be more prevalent in open ocean waters. However, if you go to other systems such as coastal waters, this is the eastern coast of the USA in the state of Rhode Island. This is an estuarine system, Narragansett Bay. Here we sampled in different parts of the bay from, along 5 months in 2017, and we found that there is like a huge prevalence of delta protobacteria. And this is interesting because it seems that delta protobacteria are always more prevalent in coastal places and gamma in open ocean places. And we think this is because of their metabolic versatility. So many delta protobacteria, besides being able to fix nitrogen, they can also denitrify. So in places such as Narragansett Bay, that is an estuarine system with lots of rivers around it, bringing nitrogen into the system, maybe it's more ecologically worthy to be able to do both nitrogen fixation and denitrification so that you can adapt to these fluxes of nitrogen. Whereas in open ocean systems such as this, where nitrogen concentrations are very low and very stable, you don't really need to have that metabolic plasticity. But this is something to be confirmed, of course, with further studies in other regions of the ocean. Another potential place where to find NCDs, and this has been something we have been working on in the past 10 years quite hardly, is the dark ocean. So if you think about the distribution of the water column, so you know that the open ocean can go down to up 11,000 meters. But the place where you have light is just the very upper layer, what we call the epipelagic zone, which goes down to 200 meters. And below that, there is basically no light to do photosynthesis. So you would expect that your cyanobacteria, because they are photosynthetic, they will be restricted to the epipelagic layer. But then NCDs, if they don't need light, maybe they can be anywhere else. So this is something we started to work on to measure nitrogen fixation rates and to get DNA samples from both the mesopelagic and the bathypelagic regions of the ocean. And without going into many details, because there have been several works on this, but pretty much the results that we get are always pretty much the same. This is an example from one cruise in the Solomon Sea. So this is in the South Pacific, close to Papua New Guinea. So we looked at the diversity of these NCDs, and we found a huge diversity, pretty much everything that you find in the different clusters of the NIFH gene. So there was a gamma protobacteria, delta, alpha, beta. This cluster 3, there are obligate anaerobes, and also these putative nitrogenases from cluster 4. So pretty much everybody was present in the samples. When we measure nitrogen fixation rates, we always measure something measurable, but actually quite low. And there doesn't seem to be any particular trend with depth or with other variables. So it's really hard to tell what the activity is responding to. We do find, however, a correlation with dissolved organic matter. And this is probably because these NCDs, because they cannot photosynthesize, they need another source of organic matter to make a living. So they are probably using DOM around them. But thus far, it has been hard to reconcile this huge diversity with actual nitrogen fixation rates, because there is currently no specific method to measure nitrogen fixation in particular groups. So you will always measure like the bulk rate, that is like the sum of the activity of every organism in your sample, and of course diluted as well by the organisms that are not actually fixing any nitrogen at all. However, something you may have noticed in this slide is that cyanobacteria were also there. Here I'm talking about samples that were collected at 850 meters, 900 meters, so very deep in the water column, and we found things like calotrychs, using A, and also Trichodesmium. So we were wondering why they were doing down there, and how is this possible that they were found at such depths where there is no light for them to live on. So this is something that we started seeing in other works as well. So for instance, this is the expedition Malaspina, where they found Trichodesmium, but also Richelia between 2,000 and 400 meters at different locations around the ocean. This is with using just microscopy. Also in sediment traps in the North Atlantic that do 3,000 meters, we find like intact Trichodesmium colonies that you can see here in like sticking to the walls of the sediment trap tubes. But not only Trichodesmium, also when you go to the Tara Oceans data and you look at other diazotrophs such as using A, you also find them in several places around the ocean at even at 800 meters depth, and you even find them in the metatranscriptome. So this suggests that they are actually active down there. So this is a question that we wanted to ask ourselves. We wanted to know whether these sinking cyanobacterial diazotrophs are actually active when they reach the dark ocean. So to answer this question, we went on a cruise in the South Pacific. This is Fiji here, and this is the Tonga volcanic arc and New Caledonia. So we sampled two particular stations where we deployed these sediment traps at 3 different depths, namely 170, 270, and 1,000 meters. So each trap consisted of four tubes, and out of these 4, we used 2. So one of them had 15N labeled brine. So this is a stable isotope that allows us to measure nitrogen fixation rate. So to measure the incorporation of 15N into the biomass of the diazotrophs. And the other tube was filled with RNA later to preserve RNA for further analysis. And so by doing so, we extracted intact Trichodesmium colonies from the traps and analyzed them on this machine that you may have heard from Rachel probably, the NanoSims, that is a single cell mass spectrometer. And we actually measure rates going anywhere from 26 to 200 femtomoles of nitrogen per cell a day. And this number may not say much, but this is actually the range of rates that we measure in the surface ocean as well. So this suggests that these organisms, even when they are sinking down to 1,000 meters, that they are devoid of light and they are experiencing the harsh conditions of the dark ocean. So this darkness, these cold waters, and as well more pressure, they are actually fixing pretty much the same they do in the surface. So this was quite impressive to see. And actually, Trichodesmium were providing some 70% of the bulk nitrogen fixation measured in every trap, meaning that there is still some other diazotrophs in the sample that are actually fixing nitrogen as well, but that we were not able to image on the NanoSims because they are probably smaller. So you need other methods to go find them on the NanoSims. So then we looked at the RNA, and we did NIFH sequencing on that RNA, and we found as well that Trichodesmium was dominating NIFH expression in the trap. So this is the green color here. So you can see that they dominated the expression of NIFH in both the stations. And also we found some other groups that are typical Trichodesmium epibionts, such as Chlorobium or Jangia. So this suggests again that these Trichodesmium were actually quite healthy and living in their typical conditions that we find in the surface ocean, but just at 1,000 meters depth. So now let me go back to my NCDs. We had a little Kit Kat parenthesis with the cyanobacteria. But again, I would like to reinforce the message that NCDs are actually much more important in or represented in databases. So we don't really know what these organisms are doing, if they are actually fixing nitrogen or not. As I said before, we don't have a method to measure their specific nitrogen fixation activity. And we don't know much because we don't have them in culture, so we don't know much about their metabolism or their lifestyle. However, we start to have some trends. For instance, if you look at phage abundance, this is a transect in the North Atlantic. The black dots represent gamma protobacterium, so this is our NCD, and you can clearly see that we find them, if we do like a size fractionation of the sample, we do find them in the 10 microns filters, in the 3 microns filters as well, but we do not find them in the smaller fraction, suggesting that they prefer to live attached to something else that is larger than them. This has also been seen looking at the metagenomes and the metatranscriptomes of the Tara Oceans data. So this gamma A, for instance, is always found in larger fractions, so up to 5 microns or even up to 10 microns in different parts of the ocean. But why would NCDs like to live attached to particles? Well, particles in the ocean can generate low oxygen cores. And this is potentially a good spot for NCDs to live on, because they will benefit from the organic matter that the particle is providing them, and reminding that NCDs that cannot do photosynthesis. And also they will be experiencing low oxygen conditions. And this is important because oxygen actually deactivates the nitrogenase enzyme. So they need to find somewhere that has low enough oxygen conditions to fix nitrogen. So cyanobacterial diazotrophs have different strategies to get rid of that oxygen, such as separating nitrogen fixation from photosynthesis along a trichome, for instance, or in between the day and the night. But NCDs, we don't really know how they get rid of that deactivating oxygen that is actually poisonous for them. And this may be an explanation. There have been some models trying to explain this process. So this would be the evolution of oxygen and nitrate in a particle along 4 days. And the idea is that you start with a very fresh particle that is very labile and very rich in organic matter. And then a first bunch of bacteria get attracted by this fresh organic matter, and they start respiring and consuming oxygen. So oxygen goes down. When you don't have, when oxygen is gone, then you're going to use the next electron donor in line, which will be nitrate. The nitrate goes down, and then a window appears where nitrogen fixation can occur. This nitrogen at a certain point starts being remineralized and going back into nitrate. So then nitrogen fixation disappears again. So this is a theory, this is a model, but this has not been proven in field experiments or in laboratory experiments. So this is where we're trying to go now. So this is my student Arthur. He's working with Vibrio diazotrophicus, which is one of the few NCD models available in the in strain collections, although it's not very representative from open ocean waters, but it is from coastal. So he has been incubating these Vibrios with and without particles, with and without glucose, to look at what they do. And first thing that we see is that they do need particles to fix nitrogen. So what you see here, it's in blue, DAPI staining, and in pink, it's an antibody labeling of the nitrogenase enzyme. So you see that when they have both particles and glucose, they aggregate around the particles, and they are expressing the enzyme. But when they have only particles, but they don't have glucose, they do so as well, but not as much. However, when there is no carbon around, when there's no particles and no glucose neither, then they just don't fix nitrogen at all. So this was very promising, and we wanted to go a little bit further. So this is very recent data from a cruise in Hawaii from this summer. where we used a device called marine snow catcher, or MC. And this is basically a very huge Niskin bottle of 100 liters, where you deploy it at the depth that you want to sample. In our case, it was 200 meters depth. Then you bring the bottle on board and you allow it to sediment. In our case, it was for 4 hours just on deck. So that will, along those 4 hours, the particles that are in your water will start separating by sinking speed. So it's a very qualitative approach, but basically we obtain 3 fractions. So the suspended particles, the particles that are just like suspended in the water column and not sinking significantly. Then the slow sinking in this lower compartment. And then at the very bottom, there is like a plate where we obtain the faster sinking particles that really reach down to the end of the bottle in 4 hours. So we took samples from each of these fractions to work on the DNA, to find the isotopes, and also to measure nitrogen fixation rates. And this is very preliminary data. These are qPCR, NIFH counts done on board during the cruise by our collaborator Kendra Trukobo. And well, we find that some of them do not seem to prefer any particular particle fraction, but however, others do prefer to be in the suspended particles, while and usually they decrease towards the fast sinking. So this is pretty much work in progress, and I'm hoping to be able to tell you more in the following months. When we look at the activity though, so this is the bulk nitrogen fixation. So again, not differentiated by the different groups. We do see quite high nitrogen fixation rates in the suspended. So these are about like somewhere between 12 and 30 nanomoles per nitrogen a liter a day. So this is actually quite high. So the typical rates that we see in the North Pacific surface ocean are somewhere around here, somewhere maybe between 2 and 10. So we were like quite surprised to find these very high rates. And they are actually also detectable, but lower in the suspended and the slow sinking fraction. And in the fast sinking fraction, we weren't able to detect them, but I do believe that it's not because there wasn't any activity, but just because we didn't reach the detection limit of the machine, because the volume that you recover from this lower part is just 300 milliliters. So that is sometimes not enough to reach the detection limit of the mass spec. So our next step now is to try to go further into those samples and to develop a specific method to measure nitrogen fixation by different groups of NCDs. So something we did during the cruise is take single particles, incubate them with 15N. You can see this little brownish little thingy. It's being incubated with 15N. And we will extract DNA from them to construct mags with the idea of designing a specific cartridge probes so that we can actually detect different groups of NCDs into the particles. So some of the particles can be quite large, and if you put them on a microscope, you're going to face topography problem, and maybe the NCDs are more towards the core, where they can obtain those oxygen-free conditions. So we are working on a protocol to embed the particle. You can see it here more or less embedded in a resin, so that then you can cut it with a microtome, so that you can analyze also inner part of the particle. So this is very preliminary work, and you don't see it very well here, but these are basically different slices of this particle here. And this is very recent work where you see again these the expression of the nitrogenase pretty much in this pink little dots, pretty much in the center of the particle, which is the whole blue cloud that you see around it. So this is very promising. So the next step, and this is the work of Christian, who's a postdoc in our group. He will combine these cuts with nanosims to actually measure nitrogen fixation rates by specific groups in different parts of the particle, with the ultimate goal being to pile up these different microtome layers to reconstruct the particle in 3D. So yeah, this is ongoing work. So with that, I'm just going to go to my take-home messages. So I hope I have convinced you that nitrogen fixation is not only occurring in the subtropical zones, but also in higher latitudes. that we are facing an expansion toward these higher latitudes in the future ocean. At least this is what prediction says towards the end of the century. That NCDs are probably impacting nitrogen fixation globally importantly, but that we don't have the means to know this or to measure this precisely, and this is what we're working on. And we don't actually know much about these NCDs and how they regulate their nitrogen fixation activity. So beyond that, I think that we need more specific methods and also high resolution measurements, because so if you think about the model that I showed you of nitrogen fixation associated to a particle, you see that it happens in a very particular window in time, meaning that if you're measuring just before that window or after that window, you're just going to miss it. So probably we need like underway methods that are able to tell us what is going on at a higher temporal resolution so that we don't miss the actual peaks. And finally, an important message as well is that we need to incorporate this full isotope diversity in biogeochemical models, because I haven't got much into that, but current models do only contemplate Trichodesmium. Some of them contemplate Crocosphaera, but there is no representation of important diisotopes such as UCNA, and certainly not of NCDs in global biogeochemical models. So this is like really affecting our knowledge of how nitrogen fixation is responding to climate change, how it is responding to ocean circulation. So this is a main problem that we need to work on. But I think before we can get to incorporating this diversity into the models, we need to understand what is happening at the cell level before we can extrapolate it into something larger. And while that said, I would like to thank you all for listening, and I would like to thank my group and of course all the funding agencies that have made this work possible. And I'm happy to take any questions.